You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health Podcast. I have Jianyi Zhang, Dr. Jianyi Zhang. He's known as Jay Zhang, so I'll call him Jay. He's an international leader in myocardial bioenergetics, biomaterials, and stem cells for cardiac repair. He's a tenured professor of medicine and of engineering, the T. Michael and Gillian Goodrich Endowed Chair of Engineering Leadership, and chair of the Department of Biomedical Engineering, Joint Department of the School of Medicine and School of Engineering at University of Alabama at Birmingham. So, Jay, thanks for coming. How are you doing?
I'm doing very well. Thank you, Richard.
So what's myocardial bioenergetics? I understand biomaterials and stem cells for cardiac repair, but what are bioenergetics?
Myocardial bioenergetics is talking about how the energy is produced in the mitochondria, transport to the contractile apparatus, and utilized in the contractile apparatus to generate work and generate force to pump the blood, support the whole body.
Huh. Okay. So I guess they used to call it the Krebs cycle in the mitochondria. ATP is produced, and then it migrates out to the proper cells that need it.
Yes. So the Krebs cycle is more in the petri dish or in the in vitro study, and my training is examining in the whole organ system in the in vivo system, using the large heart or large animal model or human heart.
So what happens, if you don't mind just giving a basic overview, when the mitochondria produce ATP? How does it get to the right cells in the heart, and how does it cause them to contract?
So this goes back to a few decades about the energy stuff, the failing heart hypothesis by our Annie proposed that heart failed to pump because of loss of energy. So this is a vague concept. In the past few decades, people have examined how the energy is produced. Mitochondria or myocyte can utilize the glucose or fatty acid to generate the ATP, and ATP is transported to the contractile apparatus to generate work. And it has been consistently found that in the in vitro, in vivo small animal models, large animal models, and the patient, we found that heart failure is associated with the abnormalities in energy utilization or energy production related enzymes. And the severity of the alteration is severely or linearly related with the severity of heart failure or prognosis of the death or the patient survive in 5-10 years. So it's severity of energy abnormality, of energy metabolism abnormality, is a prediction of the prognosis of a patient with heart failure.
So in someone that, what are the symptoms? How will that manifest? If will their heart just beat less forcefully? Will it not keep a rhythm? Like what happens, pathologically in a person?
Yeah, that's an important question. Now, heart failure is being utilized vaguely in the field of cell biologists, molecular biologists. All the scientists that deal with the small animal models. But heart failure is a symptom. It's the end stage before going death. All the heart problem before going to death is end up with irreversible heart failure, which congestive heart failure, which is a dilated heart, cannot pump, and eventually go to death, which is a trillion-dollar business to take care of this clinical problem in the world every year. $1 trillion means it starts with a T. So it's a very important public health problem, and physician and physician scientist, engineer has been working together trying to solve this problem for decades. And recently is cell therapy or bioengineered tissue to repair this broken failing heart.
So what are the stages of heart failure? How does it begin typically, and how does it not end? We know what happens when it ends, but close to the end. What are the steps?
So we have a concentric hypertrophy secondary to the pressure overload, which we usually say is hypertension. So it's a very thick wall with small cavities. So it's a classically concentric hypertrophy, a heart with increased LV mass without severe coronary artery. There's dilated heart failure with the valvular problem, so insufficiency of mitral valve or aortic valve make the valve cannot close, so it's dilated cardiac hypertrophy. The third case of compensated hypertrophy is post-infarction LV remodeling. That's what the cell therapy has been targeting on, utilize the cell as a product to prevent the post-infarction LV remodeling become heart failure. So in the clinical case, when the infarct is small, patients survived or saved at the event. And then the patient have heart with LV scar. And over the time, if the scar is big enough, it's gradually have a dilated left ventricle.
What people will people have a series of small infarctions that slowly scar more and more of the heart?
Actually, it is. That's a good question. Actually, how the post-infarction LV remodeling started was a compensated LV pumping over the time of months and years, become a dilated left ventricle with very decreased ejection fraction and end stage congestive heart failure. The mechanisms remains unknown. It's just probably multifactorial result in this end stage heart failure situation. It not necessarily have repeated small infarct. It is likely the overstretch of the border zone of the LV scar. The overstretch triggered the detrimental signaling mechanisms to make the border zone go on apoptosis because of the overstretch, because of the detrimental signaling pathway, because of the energy metabolism abnormality. So over the time, the borders will expand. The characteristic of alteration of energy metabolism and overstretch.
So what happens in a heart with hypertrophy? Like, is it that the elasticity of the ventricle changes? Is it that literally the cells are enlarged in that ventricle? Is it mechanical, where the cells have physically moved apart to create a larger ventricle that requires more pressure to pressurize it? I mean, what is the mechanism of hypertrophy?
The hypertrophy is secondary to the pressure or volume overload or the stress. So it's just like skeletal muscle. When you exercise, the muscle cells getting bigger. So in the heart, it's a bigger heart is a problem. We call hypertrophy. And
but that's because the cells are bigger, or is literally the morphology of the heart changed?
No, it is cell bigger and longer. So the normal myocyte is about 100 micron in length. The hypertrophy, the heart length increase, but more of a cross section is thicker. Cross section area is can be doubled or even tripled.
Oh wow!
Therefore, the perfusion support the mitochondria in the myocardium can be one of the factors that because the cross section area is double or triple, the diffusion of nutrients, oxygen to the mitochondria is a factor that can be compromised.
So you're saying in a bigger cell, it takes more time, or if you have a certain number of mitochondria in a given cell that's fixed, let's say, and the cell is now twice the diameter, it takes longer, I guess, for the ATP to diffuse throughout the cell to make it contract and do its job, or to, I guess, provide energy to the whole cell.
That's not that simple, actually. Exactly how the energy metabolism's characteristic contribute to the dysfunction of a pump or contractile function is still unclear. We know two factors. We know two facts. One is the heart fail to pump sufficiently. The other fact is the metabolic characteristic of the myocardium change. Whether this bioenergetic changes caused by the pump to fail, it's still being demonstrated.
So you don't know if the mitochondria are recruiting other methods, perhaps, of providing energy, or that maybe the biochemistry of the reaction itself changes somehow. I mean, there's many different possibilities, right?
Yes. So actually, that's another good question. In the heart, the energy metabolism reserve. This is a key term. People always often forget about that. The energy metabolism reserve is in human heart. It's probably 5-fold. In the different species, even higher. So there's a significant reserve. So unless you abolish the reserve, you cannot demonstrate the decrease is causing the dysfunction. So
as a heart experiences hypertrophy,
then the option of taking the substrate to support energy production is another factor. So the mitochondria can utilize glucose, you can utilize fatty acid, can utilize ketones, can utilize anything available. If you abolish one thing, they take other ones. That's why it makes the question difficult to examine and demonstrate and examine the mechanisms. So, but we are getting closer to understand the problem. We are getting closer to alter the pathways to utilize as a therapy method to prevent heart failure, to cure, to delay the heart failure to occur.
Okay. And once you experience hypertrophy in a heart, does that change the shape of the ventricles or the atrium? And
Yeah.
Do you see like a morphological difference?
Yes, it mainly changed the football to like softball. The hypertrophy heart is more round shape.
Oh, interesting.
Yeah.
So, have you tried modeling different shapes of ventricles and, pumped some fluid through them to see how that changes the fluid dynamics just because of the shape?
So over the past decades, people have tried different ways. Surgeons actually mainly tried different ways to use change the shape or limit the dilatation to prevent heart failure or improve the pump to pump the blood. One is it's called LV resection. So the South America surgeon tried to in the open chest surgery to cut pieces of the heart and suture together to prevent a heart failure to improve the heart pump. It didn't work. The other trial is try to put a mesh to outside the ventricle to prevent dilatation, to prevent change to a softball shape. We called it a condom for cardiologists. It didn't work either. This is just two examples of different intervention to change the LV shape to prevent heart failure. It was not successful. Basically, it is a end stage of a disease. Actually, it's a syndrome. It's many different heart disease. The end stage, everything, all the original origins of a cause of the disease end up with the end stage heart failure with a dilated heart. And next step is death.
Okay. So what area of all this are you trying to figure out? I can see now, I understand there's so many different mechanisms going on. Maybe certain, epigenetic changes are changing the gene expression, which changes how the heart contracts and, how it metabolizes energy. I mean, it could be many things, I guess.
Yeah. So by training, I'm trained as a cardiovascular physiologist. So I know the heart is an organ that is different from any other organs in terms of it's a high pressure, high flow organ. Every minute it beats 100 beats per minute. Pressure increase from 0 to 100 millimeter mercury 100 times every minute, and every day it utilizes 10 kilogram of ATP and pumps 10 tons of blood. So it's a very, very tight structure. When we try to do the cell therapy, we inject cells into the myocardium. The cells face this cyclic pressure increase and decrease. Therefore, that's one reason the engraftment rate is so low. That's remain to be one major problem of the cardiac cell therapy, low engraftment rate. Even though with the autologous cell therapy, is also in these cells, transplant cell encounter this cyclic pressure increase, decrease, and high flow, high pressure organ. So I started early by try to fabricate engineered myocardial tissue, like patch a engineered tissue on the surface of the LV injury, so the cells are not facing the cyclic pressure increase and decrease. So it's a better environment, direct contact the LV injury. So that comes with the cardiac muscle patch therapy. It tends to have a better engraftment rate by allogenic or the xenogenic transplantation approach. But the engraftment rate remains in the single digit. So it's prevent LV dilatation. Dilatation is a major problem. If we can prevent the dilatation, it's effectively help the pump. So with the biological active cell patch on the surface of LV injury, it effectively prevent LV bulging. That's many of the beneficial effect based on, in my mind. Going forward, I think to promote graft myocyte proliferation is the key. So if the engraftment rate is single digit or in the 0.1 to 0.9% of engraftment rate, if we can make these myocyte graft less than 1% of myocyte proliferate, if we can turn back the clock of myocyte, go on proliferate, then this compound principle, in other words, cell dividing 1 to 2, 2 to 4, 4 to 8, over the time, even though it's a 0.1% of cell graft, over the weeks, we can easily get the millions and millions of cell myocyte to replace the lost myocardial mass during the heart attack. So my focus has been focused on the promoting myocyte proliferation, or otherwise turn back the clock of myocyte proliferation. As you know,
I'm not sure if I understand. What do you mean myocyte proliferation? Like, what do you mean? Like, when the patch is applied, only a very small percentage of the cells stay active, or what? Like, what happens?
We need to go back a few steps to the developmental cardiovascular biology. So the heart, before prenatal stage, heart myocyte continue to proliferate, meaning the myocyte double, the cell doubling time. Myocyte continue 1 become 2, 2 become 4. That's hard form in the developmental stage. It continue to divide. At the time of a birth, for whatever the reason, when the organ exposed to high oxygen start to work, the myocyte exit cell cycle. So within a day or maybe 3 after the birth, myocyte exit cell cycle, meaning they no longer divide. So you born with a fixed number of cells, you die with fixed number of myocyte. That's why heart attack become such a serious problem. When you lost 10% of myocyte during heart attack, you survive with a big piece of scar. If it's greater than 10%, many cases it's died on the site. So myocyte do not regenerate. It's the major problem of a heart attack or heart failure. So if we can find the regulators of how the regulators control the myocyte exit cell cycle within 3 days after the birth, then we can manipulate this regulator to turn back the clock, to make the cell proliferate. If we can control the cell cycle, then we can likely prevent heart failure or cure the heart failure. Because basically heart failure is a problem of heart lost the myocyte. Myocyte is the basis of the cardiac pumping.
So you're saying that, all right, so in a heart attack, myocytes, a certain percentage of myocytes die. We don't make any more. So the existing or the remaining ones enlarge to try to accommodate. Is that the process?
Yes. The compensate, it's called compensated hypertrophy.
Okay. And then once they do, how does that lead to dilation and then heart failure?
Yeah.
Once the remaining cells grow, what happens to them?
What happens is multifactorial. It's related with energy metabolism alteration. It's related with the other signaling pathways also. So it's still not clearly defined. It's a multifactorial event to make this happen, from compensated hypertrophy to heart failure. But if we can prevent this dilatation, prevent the further hypertrophy, compensated hypertrophy is not at the stage of severe dilatation. If we can prevent the dilatation, then it will prevent heart failure to occur. That so far we have not been successful.
As hypertrophy continues, it leads to what? What it just keeps going and going, and the cells get bigger and bigger, or what is the course of hypertrophy?
Elongation. It's the elongation. The 100 micron in length become 200 micron in length. Eventually, they fail to contract. The contractile apparatus have a fixed length. If you overstretch it, it will fail to contract.
So that wouldn't that wouldn't that change it changes the contractile strength?
Mm-hmm.
Would it change the heart rate? Would it change other factors about the heart?
It will not change the heart rate. The electrophysiology is a separate field. The hypertrophy, severe hypertrophy is associated with increased sudden death, meaning arrhythmia related. But how exactly a heart without coronary artery, severe hypertrophy, the sudden death rate increased the fivefold. It's complicated also.
So does science even understand the mechanism of why hypertrophy occurs?
That's known, actually. Hypertrophy occurs as the stress increase. So either pressure overload or volume overload at a cellular level is increase the stress, meaning either stretch or contract more contraction. So it's just like in the gym, you exercise more, the more hypertrophy. That is known. But how hypertrophy leading to the heart failure or leading to increased sudden death, that is more complicated. Why is myocyte hypertrophy? This is a more simple question to understand, to explain.
Okay. So what happens when you apply? So you're culturing cardiomyocytes, and you're encouraging them to go back to a stem-like nature. You're culturing them in a dish, and then you're applying them as a patch to the patient, or what's the process there?
You are talking about the engineered tissue, or in other words, fabricate human cardiac tissue in the petri dish and utilize that engineered myocardial tissue patch on the surface. Right. It's basically a simple, intuitive approach. So you have a heart survived from heart attack, lost 10% of LV mass. If we can create a muscle patch onto the surface of this LV injury, if we can make this muscle patch, engineered human muscle patch, survive on the surface of the injured heart, can effectively prevent the LV bulging or overstretch or dilatation, then you likely can prevent heart failure to occur.
Well, why do you think, why is the left ventricle more susceptible to this than the right ventricle?
Susceptible to the heart attack?
Susceptible, I don't know. Is it more, when heart attacks happen, do they tend to happen on the left side of the heart or the right? Where do they happen? And then hypertrophy, where does that happen? And then, heart failure, where does that happen?
Yeah, right heart or right ventricle and left ventricle, they both can have hypertrophy secondary to different disease origins. The reason left ventricle has been examined much more than right ventricle is because the left ventricle is the pump to pump the blood to support all the important organs in the system. Right ventricle is supporting the lung, we call the low pressure system, and the left ventricle is a high pressure system. And heart attack and the left ventricle often have the causing the heart failure and causing the clinical problem is also because it's the pump support all the important organs.
Okay, so do heart attacks tend to happen more on the left side versus the right?
Yeah, yes.
Okay, so higher pressure, more problems. Okay.
It's the pump. If the left ventricle fails, the system fails. The right ventricle fails, the lung fails.
Right. Gotcha. So your focus is you're trying to understand how cardiomyocytes proliferate in an embryo in the beginning, and then they stop. So what is your vision of a goal?
So that's the two ends. One is for engineered myocyte tissue patch. I want to have the myocyte that they utilize to engineer the muscle patch. There's increased myocyte proliferation. So same 0.1% of graft rate or 1% of graft rate. If the muscle cells in the patch, they can continue to proliferate. Then for the purpose of muscle patch, prevent LV bulging is more successful because you basically end up with larger and thicker graft to prevent LV dilatation. That's from the transplantation perspective. From the prevention of heart failure perspective, if we can make the recipient heart myocyte go back to the clock, turn back the clock, start to proliferate when there's an injury, then the proliferation can fix the 10% of LV mass loss during the heart attack, which is my hope. We can figure out the regulators, utilize the modern technology of bioinformatics, large data to find out the regulators. It will not be one regulator. It will be multiple regulators. And then we can manipulate, use the modern gene therapy technology to turn back the clock of recipient myocyte. Then they start to proliferate. If we achieve that goal, then they can one day to remuscularize a injured ventricle. The key here is to remuscularize a injured ventricle, injured during the heart attack.
What do the patches do right now? Do they work? Do they fail? Are they barely workable? Like what happens when you put a patch on somebody?
It's very exciting and quite promising. We recently published a paper in circulation that fabricated engineered human cardiac muscle patch that is thicker and larger. It's clinical relevant size. Have the in vitro fourth generation, one of the best. It's about normalized about 1 nanonewton per myocyte, which is quite encouraging and promising. So if and let's see, we engineer this myocyte with cyclin D2 overexpression. Cyclin D2 is one of the key regulators, regulate the cell cycle. So these myocyte have a higher proliferation rate in vitro and in vivo also. 4 weeks after the transplantation, the engraftment rate is significantly higher. So this approach of utilize myocyte was engineered gene expression, turn back the clock. It likely will have larger graft. In other words, remuscularize injured ventricle, which lost myocyte during the heart attack.
So, but again, once you apply the patch, what happens? Do those cells die? Do they scar up? And only a very small percentage of them make it? Like what happens?
Yeah. So at this point, we have tried catheter-based vessel delivery of the cell therapy. We have tried direct myocardial injection of the cells to the myocardium, or we try the patch. It's all at the rate of single digit. 4 weeks after the transplantation, whatever the means to delivery, it's always maintained the single digit or less. Meaning, again, go back to the beginning of our conversation that this is because the heart is an organ that are not welcome to any foreign stuff because it's a cyclic contraction and high flow, high pressure. So, however, if we can make these 1% or 0.1% survive the cells 4 weeks after the transplantation, continue to proliferate, then it's a simple compound mechanisms. A few weeks further, you can easily generate millions of myocytes by simply successfully turn back the clock of myocyte proliferation.
So you're saying only a single-digit percentage of the myocytes in a patch survive, and how fast do the rest die off?
It's about 10%. Yeah,
how fast do the rest die off?
The rest just die off.
How fast?
I'm sorry.
How quickly does that happen?
It's in the rule of thumb. It's the first week probably lost more than 70, probably more than 80%. Majority lost. In other words, majority lost was in the first week.
So, is it? Do you think it's because of an immune response, or is there a cell-to-cell communication between the new myocytes and the existing ones, telling them to you're not needed? I don't know.
Yeah, the immune rejection is one of the major causes of the low engraftment rate. So, which can simply demonstrate by if you look at the rodent study with immunodeficient rodent, the engraftment rate usually is much better than the pig or dog study because we just don't have immunodeficient pig or dog study, dog models, immunodeficient pig models. It's too expensive. Nobody have utilized to study the cell therapy yet. Most of the immunomodulation, immunorejection are studied with the immunodeficient mice compared to wild type. So immunorejection is one of the reasons that they lost the cells in the first week.
Are these the patient's own cells, or where are the cells coming from?
That's autologous cell type. It can be done with the animal study. For example, in early days we have done the pig study using the bone marrow derived mesenchymal stem cell transplant into the recipient heart with heart injury. The engraftment rate is also very low. So it's that will indicate other than the immunorejection, the other reasons will cause the cells not graft into the heart.
So it sounds like we can't take existing myocytes and induce them to be pluripotent stem cells and then culture those. It sounds like that's not possible.
Say it again.
Can you take existing cardiomyocytes and induce them back to pluripotency? Can you make them stem cells again from mature cardiomyocytes?
I think technically it is doable nowadays. Any cells with nuclear, we can reprogram into the stem cell, and then can differentiate the stem cell to any other cell types. Myocyte is a special case because it's larger, much larger, and has multi-nucleus in each myocyte. So nobody has tried making the myocyte become a stem cell. It's
right.
Yeah, it's a study, but the turn back the clock is not changing myocyte become a stem cell. It's changing myocyte genes characteristic to make it back to the embryonic stage of continue to proliferate. We don't need to go back really to embryonic stage. We just promote the promote the cell go back to the cell cycle, meaning proliferation.
Do you do this with the patch cells?
Yeah,
or you would do this with existing heart cells?
We actually have done one study that's the cyclin D2 overexpression study. We successfully make a much larger graft in 1 week, and in 6 weeks, in 6 months, it's actually repopulate 50% of LV scar, which has not published yet. But remuscularization with promoting the myocyte cell cycle can result in remuscularization of injured ventricle.
So was it 50% of the patch cells surviving, or were they creating new cells from the patch cells? Like, what was the mechanism, you think?
It's the proliferation of a grafted cell.
Proliferation meaning new grafted cells were being created?
Proliferation means one cell become two cell become four.
Right.
So the seed cell is not the recipient myocyte, it's the graft or the transplant cell.
Are you cutting out the dead tissue and then replacing it with the graft, or are you putting literally a patch over the dead material?
Yeah, patch, the engineered muscle patch on the surface of LV injury. We are not cutting the...
So you're literally patching over dead tissue or scar tissue.
Exactly right.
So, all right. Can you tell with the patch, you said most of the patch doesn't survive. Where doesn't it survive? Like, I figured you would anchor it. I'm just going to pretend it's like a circular patch. You know, you'd anchor the patch at, let's say, 50 points somehow around the circumference of the patch, and then it would float or, be attached but kind of float on top of the damaged area. And if so, what part of the patch survives and what part dies? Is it uniform that only a small percentage survive, or are there certain areas of the patch that survive better than others? Like maybe the connection point survives, but the middle doesn't, or vice versa.
So right now, in the immunodeficient rodents study, the patch, because of the rodent does not have the immunorejection, so the engineered muscle patch can survive onto the surface of the LV injury. The loss more or less is the uniform. It's about 10% of the myocyte survive. The significant improvement of graft size is again by the promoting the survive the 10%, continue proliferate. By the time of the 6 months, it repopulate the LV scar. So that is achieved. We have not talked about another question, that is the resupply the graft, the circulation or the vascular large grafts. So the muscle graft need a high oxygen and chemical energy to support, to function. And how the large graft, if we lost 10% of muscle, we regenerate 10% of muscle. How this 10% of muscle mass is supplied by the perfusion? That's an important question for the field. So the key here is not the pre-vascularized engineered vessel. It is how the engineered muscle can survive for the first few hours or first 3 days. If the muscle can survive for the first 3 days, the angiogenesis, meaning the sprouting of pre-existing vessel, can penetrate the fibrotic tissue and get into the graft muscle, can supply the oxygen and carbon substrate need for the grafts. So it's not a growth vessel issue. It's how the muscle, engineered muscle, can survive for the first few days. That's the most difficult part. And
do Do you see any difference in larger versus smaller patches? Do smaller patches survive longer, or no difference?
We can make a multiple smaller become larger, but the thickness is the key here. So the one we recently published in Circulation is about 1.2 millimeter thickness, which is probably the best so far in the cardiovascular tissue engineering, 1.2 millimeter again. So it's still relatively thin, but it's the best so far in the literature. So how do we engineer such a tissue in the petri dish? It's basically a dynamic culture, meaning the cell need to have a physical environment. In other words, it's a mechanobiology. Cell, if you just put in the 2D culture, they do not function as well as if you put it in the dynamic culture. That means cell membrane interact with the moving medium, change in physics in the environment. So the dynamic culture make it a significant better function and a thicker engineered tissue patch. That's why it was in circulation.
I gotcha. Well, Jay, there's many more questions I could ask you.
Yes.
But, we're almost out of time.
Mm-hmm.
What's the best way for listeners to find out more and read some of your papers and to get in contact?
We have a symposium for the specific cardiovascular bioengineering to use the pluripotent stem cell to engineer the cardiac tissue or promote cell therapy. This symposium has been going on for the past 5 years annually, and with the world leaders participate. It's funded through the NIH called the PCTC program. It's a progenitor cell progenitor cell translational consortium. This is the 10th year funded through NIH, and the meeting is participated by leaders in the field, in the world, and every year after the meeting, we work together to generate a statement or a review, which has been published in the Science Translational Medicine or in JACC, Journal of American Cardiology College, JACC, or circulation research. So that's the area you can find our my work and our colleagues' work.
Yeah, the website seems to be translationalcells.org. Looking at it now.
Yeah, it's a PCTC. If you search Google the NIH PCTC, will lead you to the website. And you can search the UAB BME. That's all these people's lecture is actually on the website.
Okay. Well, very good.
Mm-hmm.
Well, Dr. Jay, thank you so much for coming on the podcast. I appreciate it.
Thank you very much, Rich. Have a great day.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only, No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.